Thank you, Ikai. Good morning, everyone. Um, Today I will present the work we did here in my lab in the last about 15 years. So I try to summarize the work from the early days till very recently. And I will talk, as Itai says, about the regulation of leaf hydraulic conductance. Maybe I will start by explaining what is this term. So when we look on plants doing transpiration, we all know that. So water being absorbed from the soil via the root, through the xylem, through the vascular system, and in the end, they are being transported via the stomata. These water are mainly used in the photosynthetic pathway. So CO2 being absorbed here in the via the guard cells, being photosynthesized in these mesophyll cells. In order to do it efficiently, these cells need a lot of water, and all this water coming from the vascular system. One issue here that we have to remember is that most of these water being lost via the stomata, so we have huge loss of water which are not used to photosynthesis at all. If you want some numbers, one banana transpire about 100 liters of water. So the production of one banana, you need to irrigate the plants in about 100 liters. To get 1 kilo of avocado, the plant will transpire 500 liters of water. So many of the studies work on these guard cells, which are very important to regulate the water. But today I want to talk about something a bit different. I want to talk about the leaf hydraulic conductance, which means the actual liquid water that move via the xylem system as a liquid phase. So I'm not going to talk, or almost not going to talk, about the stomatal conductance, but on the leaf hydraulic conductance, which means basically how water go so well through meters and meters long of vascular system. The terminology of the leaf hydraulic conductance is K-leaf, and I'm going to use this terminology a lot. I just want you to remember, we do talk about water movement as a liquid and not as gas, as usually people talk about gas conductance, okay? So what regulates this water conductivity or water conductance in the plant? Basically is the hierarchic structure, okay? The hierarchy of the vascular system has a big impact on how water flow. The more wide the vessels and the more angles you have, this will dramatically impact the flux of water through the system. So it's pretty amazing. If you look on 1 centimeter of Arabidopsis leaf, you will find about 20 centimeters of vascular system, enabling water going each and every cell in the leaf. So of course, this is very important to regulate the water, but this does not explain the dynamicity we find. So if we look on the fast changes, when I talk about fast changing of water flux, I talk about minutes of changing. When we look on this, we find that the hydraulic conductivity is very fast. Look at this paper published by Marte et al. They expose plant to drought. You see here, at day zero, the plant started the drought treatment. Now you can see that the plants reduce about 50% of transpiration in 4 days. But when you look on the hydraulic conductance, you find it was twice as fast. So plant reduced hydraulic conductivity in 2 days. Other paper by Sack and Sokofoni show that if you measure the same leaf, in this work, they measured the actual same leaf, they saw that the hydraulic conductance, what you see here in this graph, was much faster reduced compared to the stomatal conductance. So the same leaf, when exposed to degradation of water potential, this is a stress leaf, closes stomata, but much before reduces hydraulic conductivity. This is a matter of minutes. So we can say that this vascular system, which is basically made of dead cells, is important to regulation of the flux, but the dynamicity, this fast increase or decrease of water flow to the leaf, must come from a different system. And we think it's come from this bundle sheet layer, from these cells that cover the whole xylem system. You can see them here. And our working hypothesis during the last decade or more was that these living cells, these are parenchymatic cells covering all the xylem in the plant. These cells are actually important to regulate the K-leaf, which means the water flux outside of the xylem cells to the leaf, and also important in absorbing mineral to the leaf. What is the rationale to this hypothesis? So first, when you check in each plant you look, you always find these parenchymatic cells covering the veins. You always see bundle sheet. When you look on each cut and cross, you will find, you see these are the bundle sheet and these are the xylem. Each xylem cell is always in some connection with a bundle sheet. So the bundle sheet are tightly covered the xylem on any leaf you look at. It's not just the C4. We talk about C3. All these pictures I show you now are C3 plants. The bundle sheet in C3 plants does not do photosynthesis as in C4, but we think it's very important to the regulation of water. And with this question, my former PhD student, Arava Shatil, which is now running the lab of molecular cloning in Tel Aviv University, she decided she will check this question. And first of all, she had to show that if we write with this hypothesis that these cells, this bundle sheet that you see here, really control the flux, the outflux from the xylem, we have to prove that this apoplastic connection between the cells are not significant, because here you cannot control the water flux. This is passive pass. So Arava did few experiments. First, she used some dyes, and she saw that the dyes goes, these are apoplastic dyes, chalcofluor or CW, I will call it. The dyes go very efficiently through the xylem system, and in 1 hour, you see the whole leaf full of color. Even if you leave it for 48 hours, you don't see leakage of the color to the outer layers. So outside of the xylem, you don't see any contamination of the color, but you see these dots in the surrounding of the leaf, which actually represent the guttation drops. So we can clearly see that color goes in the leaf, but does not go outside of the xylem. Now, when you peel the epidermis and put a drop on these cells, in minutes you can see the dye do coloring these cells. So these cells can be colored, but probably the color does not go off. When Arava check plants that do not gutate, so these are normal plants that have gutation every morning, but some mutants cannot gutate because there is some hydraulic failure along the pathway of the vessels, and then the pressure, instead of releasing gutation drop, actually flooding the mesophyll. This mutant that could not do gutation also could not transport the color as nice as you see here. And actually, instead of seeing a gutation drop, we see all the colors spread between the vein. So if you have some kind of hydraulic failure that cannot support this pressure, everything just go without any kind of filtration, just go out of the xylem. Moreover, when Arava fed the leaf with the toxic amount of boron, which is toxic in high level, she found the normal toxicity pattern. You see, toxicity of boron is in the rim of the leaf. Although the boron goes from the main vein out, it does not toxify the middle part of the leaf, just the rim. But when you work with the mutant, you can see that wherever you have a flood, you also have a toxicity pattern. Actually, Amnon Schwartz in his lab also shows something similar. When they feed a leaf with potassium chloride, they see that the guttation drop of banana leaf actually almost did not contain any potassium. But when they feed the leaf with NaCl, so the sodium was always in the guttation drop. So the conclusion are that the banana shield has a very selective barrier that can control even ions and water. In order to continue the study, Arava had to find a way to label these cells in order to check if the aquaporins in the membrane of these cells, aquaporin are membrane protein that enable water go through the membrane in a very selective way. So in order to measure this aquaporin activity, Arava had to find a way to actually separate these cells and measure them. Luckily, we could find that the scarecrow promoter, okay, labeling or expressed only in the bundle sheet of the leaf. This is was very nice result that we could express GFP, green fluorescent protein, only at the bundle sheet. In this way, we can extract the cells and take the protoplast and use this hypotonic wash. You can watch this cell. These cells express, these are cells from the bundle sheet, and this one, you don't see it here because it doesn't express GFP. Now in this short movie, you see the cells exposed to hypotonic shock, and these cells swells while these do not. So by monitoring the swelling rate of the cells in response to the change of the osmotic value, we could use a software written by Professor Nava Moran that calculate for us the PF. PF is the osmotic water permeability, or in simple word, how fast water goes through the membrane of the cell, taking in account the changes in the osmotic concentration of the bath and the swelling rate and the size of the cell. So using this method, Arava could check protoplast taken from the bundle sheet exposed to drought. You can see a nice significant reduction in the osmotic permeability. So these cells reduce the water flow under drought, also reduce the water flow under ABA treatment. And in response to mercury chloride, which is a blocker to aquaporin, again, we saw that these cells reduce their permeability, water permeability. When we wash away the chloride, the same cells regain their water activity, which actually prove it's a specific blocker. So using the same toxin on the whole leaf, when you see this image, it means we use the whole leaf. Arava could measure the K leaf, and I want to remember, K leaf is the water flux through the veins to the leaf, the radial flux. She saw that either using ABA or mercury chloride, the cells significantly reduced their hydraulic conductivity just by feeding the leaf. When she smeared this material on the leaf, she could not see this reduction in K leaf, which means only when the cells are exposed from the vein outside, probably the bundle sheet, only then you can see this reduction. When you check transpiration, you see much smaller reduce in the stomatal conductance by the vessel feeding or by the perfusion, while you see a significant reduction when smearing the leaf. So this result is summarized in this cartoon. What we suggested in this paper is that when ABA goes in the xylem, the level of ABA actually reduces the PF of the bundle sheet cells. Less water can go through the vein to the leaf, while at the first minutes, the stomata is still widely open, which rapidly reduce water potential and relative water content in the leaf and result in closing the stomata. So here we show how a non-direct connection between the vessel and the stomata can work. And in fact, this paper was later highly cited about this mechanism. What we claim in this paper basically is that the bundle sheet is really isolated, that it's sensitive to ABA, and that we see a possible activity of aquaporin. The problem, we did not prove this water passage are real aquaporin. We only use blockers. So in order to measure if really these bundle sheet cells really have aquaporin that regulate these cells, another PhD student in my lab, Nir Sadeh, now is in Tel Aviv University on a tenure track, he decided to knock out a large part of the aquaporin family, specifically in the bundle sheet. So aquaporin family actually has few branches. This PEEP belong to the plasma membrane. Okay, these are plasma membrane aquaporin. These tips are tonoplast aquaporin. SIPs belong to the ER, and the SNPs are all over the plant, in particular in the roots. Nir decided to silence as much as possible of these plasma membrane aquaporins, and for that he built a general microRNA that can actually tackle several aquaporin genes, and he specifically expressed them under the scarecrow promoter, which means he used the scarecrow promoter. Under it, he put the MIR gene with the aquaporin MIR, and he expressed it specifically to the bundle sheet. And what we found is 8 out of 13 aquaporin was significantly reduced, specifically in the bundle sheet. This was very nice because we can also measure a significant reduction in the osmotic water permeability of these cells, as well as significant reduction of the K leaf. So indeed, aquaporin in the bundle sheet has a specific role in controlling the water outflux from the xylem to the leaf. Nir also overexpress using the scarecrow promoter aquaporins from tobacco, and it could show that overexpression aquaporin here actually increased the K-leaf. So all in all, we concluded that actually the relative water content of a leaf is not regulated only via the guard cells, but in fact, it's a combination of regulation of vascular system outflux and guard cells. These are independent mechanisms, one regulating the water flux to the leaf, the liquid water flux to the leaf, and the other regulate the gas outflux from the leaf. And together, these two mechanisms actually regulate the leaf water content and the leaf water potential. So these two mechanisms was published in few papers, and later we decided to study better which genes are expressed in the bundle sheet. For that, a former student in my lab, Noah Vigoda, today she's working in the bioinformatics department in Weizmann Institute. She worked on the transcriptome of the bundle sheets versus the mesophyll cells. And very interestingly, she found a very unique pattern of expression in the bundle sheet, which was identical to the expression pattern in the guard cells. I want to remind you all the stomatal blue light signal transduction working in the guard cells. The same signal transduction was found in the bundle sheet. So what do we know about the guard cells? We know that blue light being sensed by the photoreceptor, PHOT1 and PHOT2, start a signal transduction that activate the AHA pump, the proton pump, which pump out proton to the apoplast. And this is a very important pump because of two things. First, the pumping of proton change the acidify the apoplast, and also hyperpolarize the membrane. Due to the proton extraction, two things are happening. We have an acidic pH in the apoplast and a hyperpolarization of the membrane. These two are very important in order to regulate many membranes, proteins, which in fact, in the end, open or causing stomatal opening. So what this mechanism doing in the bundle sheet? This was the main question. So this pump, as I told you, is usually using ATP to pump proton to the apoplast. This pump is highly expressed all over the Arabidopsis plant, the AHA2. But what was nice in Noah's result, that she found that in the bundle sheet cells, this pump was expressing 3 times more than in the mesophyll. This very interesting finding actually suggested a new research which was led by Yael Grunwald, just recently finished her PhD in my lab. And Yael's hypothesis was that the bundle sheet AHA2, which is the ATPase, actually control the xylem sub pH. So we suggested that the xylem sub pH is regulated by the AHA, and the rationale for this was that we know that in the xylem, the pH is usually a bit acidic, like about 6, but either in response to ABA, you can see it in this paper, in this graph, you can see a fast alkalinization of the pH in the xylem, and the same response we see in response to drought. So these plants are exposed to drought. The lower the number, the more droughted the plant, and you see the same thing. The pH go from about 5 to about 8. So either response to ABA or drought, probably both in response to drought, actually alkalinize the pH in the xylem. So the question that Yael wants to study is, first, do AHA2 regulate the xylem pH, which means the all vascular sub pH? And second, what is the pH role in regulating the K-leaf, the hydraulic conductivity? So Yael and Noah together developed a new method to measure the pH in the xylem using a specific ratiometric dye, and they calibrated in a way that we can actually know the pH just by looking on the leaf via the microscope. So first, Yael measured the xylem pH of control plant, and again, and as expected, she get pH of about 6. But when she treated this leaf with fusicoquin is a very strong activator, sorry, of the AHA2. It's a very famous activator of the AHA2. She could saw that when the pump is highly working, we have a very acidic pH in the xylem. It reached almost 4, which is very acidic pH. When Yael uses Vanatat is a blocker for the AHA2, then we got alkalinization of the vascular system. Moreover, when Yael worked with the mutant of AHA2, she used 2 known mutants, AHA24 and AHA25. Both mutants show xylem pH of more alkalinized than the control, which means really that AHA2 is strongly regulating, or this suggests that AHA2 is strongly regulating the pH in the xylem. To prove that, Yael overexpressed AHA2, the function of AHA2 in the AHA2 mutant. You see here no expression at all. So Yael specifically complemented the AHA2 using the scarecrow promoter. And you see that in both these independent lines, she could regain the activity of the pH in the vessels. So by regaining the AHA2 just to the vascular system, these plants are still the mutants. So only in the vascular system, this plant now expressing the AHA2, and we have a very nice gain of function. So altogether, we could prove that the AHA2 is really regulating the xylem pH. Sorry. So what about the connection between the pH and the K-leaf? How is the pH related to controlling the K-leaf? So what Yael did now, she measured the K-leaf, the hydraulic conductivity of this AHA2 and 5, and she really found this plant has lower K-leaf. So something in the acidification of these leaves also reduced the K-leaf. When she worked on the complemented plant, this plant had a very normal, a normal activity of the K-leaf. This was both in the detached leaf and the intact leaf, the same phenomenon. When Yael took leaves and just regulate the pH by using, in the tube, she just put a buffered pH or non-buffered pH, she clearly saw when the pH was below 7, a strong reduction in the K-leaf was measured. Yael also took the bundle sheet cells, the protoplast, and measured the protoplast PF, the protoplast permeability, and indeed she found that these cells almost did not transport any water in alkaline pH and had a maximum activity and pH about 6. So also on the cellular level, the water did not go through the cells. So we conclude in this paper that was just recently published that actually the bundle sheet ATP as activation change or acidify the xylem pH. So the xylem pH is actually regulated by the AHA sitting here on the membrane and throwing protons into the xylem. This acidification changed the osmotic water permeability of these cells, which allow more water to go through from the xylem to the leaf, and then improve the water potential and the photosynthetic activity of the whole plant. Moreover, in the transcriptome made by Noah, we also find the FOT1. Just to remind you, the FOT1 is the blue light receptor that actually starts all this signal transduction. So now we know that AHA in the bundle sheet had a very, very important role. The question is, what is the role of the blue light receptor FOT1 that we found here in the bundle sheet expression? So here we decide to check, is the blue light involved in the K-leaf regulation? And the rationale to this question was that from the literature, we found that either in a very short induction of blue light to a leaf, very fast, this is a matter of minutes. Okay, you can see it in few minutes, a blue light induction, it starts here, increased dramatically the leaf hydraulic conductance. So leaves are known to be sensitive to blue light, which means they very sharply increase their hydraulic conductivity in response to blue light. So Yael, together with a PhD student in my lab, Sanbon Goza, they decide to tackle this question, and they build a unique chamber that can actually induce either blue and red light, blue light or just blue and red light, and expose the plant to this either red or red plus blue light. First, they check the relief, and indeed, when you expose wild type to few minutes, it's actually no more than 5 minutes of induction, you get a very significant increase of K-leaf just in response to light. When you work with the FOT1 mutant, so plant that the FOT1 does not function, this plant does not sense the light, okay? So they do not change the hydraulic conductivity based on light. The same thing happened with FOT2. And when you work with the W mutant, it's even lower than the both. So blue light does change the Arabidopsis K-leaf. When we work with the FOT1 complemented plants, so when Sanbon overexpressed the FOT1 under the scarecrow in the bundle sheet, this means these plants are the mutant. These are the FOT1 mutant expressing FOT1 only at the bundle sheet. We had a gain of function, so this whole plant continues working like normal plants, just by fixing, if you want, the FOT1 receptor just in the bundle sheet. Moreover, by silencing the FOT1 in wild type, so you took the when we take the wild type and silencing the FOT1 using the mere scarecrow, so only in the bundle sheet, we get a significant reduction of the blue light sensitivity, so significant reduction in the K-leaf. And when we use the tyrosine is a blocker that actually, it's a kinase inhibitor that actually blocks the blue light sensitivity almost at the same place where the blue light FOT1 appearing. So actually using tyrosine, you can say that blocks the FOT1 activity. So when we use this tyrosine, we could see that by feeding the leaf with tyrosine, we lose the blue light activity. Okay, this is control and this is tyrosine. When we spray the leaf with this tyrosine, the K-leaf was not changed. So only when we feed it via the xylem, we get this reduction in K-leaf. Moreover, when we check the stomatal conductance of this fed leaf, we did not get reduction in stomatal conductance, but the spray leaf did. So this shows you that when you feed the bundle sheet cells, they do block the FOT1, which does change the hydraulic conductivity. Moreover, Tanmai Torn, a PhD student in my lab, jointly guided with Professor Nava Moran, she measured the membrane potential of these cells. And by measuring the membrane potential, she could show, Tanmai showed that by exposing the plant to blue light, the cells to blue light, we get significant hyperpolarization. So blue light actually hyperpolarize the membrane. This shows you a clear activity of the AHA. But when you use a red light, in fact, you don't get this hyperpolarization. And when you use the tyrosine, you even get a depolarization of the membrane. So we continue checking this blue light impact, this time on the AHA2 mutant. So these plants are have a normal FOT1 receptor, but the AHA is damaged. In this case, again, we could not get any impact of blue light, not because of the FOT1 receptor, but because of the pump, the AHA pump. And when we use a recombinant plant, when we regain or overexpress the AHA2 just in the bundle sheet, we gain much higher or significantly higher response to blue light. Also, when we check the xylem pH, when we take and measure the xylem pH in response to light, so these plants are exposed to dark. This is night, normal night. So during the night, we have a pH which is a bit more alkalinized than during the day. Just after exposing to the light, the plants are fastly reduce their pH in the leaves, in the xylem, but they do not do it in the FOT1. So when the plant does not sense the light, they do not change the pH in the xylem. And the complemented plant does do it. So also normal night day change this xylem pH. And when we measure the protoplast, the bundle sheet protoplast, again, we get the same phenomena of if the cells do not expose to light, so if the cells are in the dark, we get much lower water permeability of the membrane than cells, protoplasts, that expose to light. So all in all, we concluded that the signal transduction pathway in the vascular system, okay, which regulate the K-leaf, goes as follows. Blue light induction actually been sensed via the FOT1 receptor, which start a signal transduction pathway activating the AHA2. The AHA2 use ATP to pump proton to the xylem and acidify the xylem. And the acidity of the xylem actually open aquaporin in the membrane, in the bundle sheet membrane, which in fact increase the K-leaf and allow water to go from the vascular system to the leaf and then outside of the guard cells. But this is only under well irrigation condition. Let's go back again and talk about stress. What happened under stress? So under normal condition, this is the mechanism we just showed. AHA2 activates pumping away proton to the xylem. But when we have a stress condition, for example, drought, then we know that the pH in the xylem is actually getting alkalinized. This is well known from the literature. So we thought that maybe this is due to inactivation of the AHA2, and we thought maybe this could be related to ABA. So maybe ABA that goes up in the xylem, blocking the AHA2, which actually alkalinize the xylem pH. This was our hypothesis. And the rationale to this hypothesis come from many papers showing that ABA goes in the xylem is the first thing that happened under stress. So xylem ABA is the first thing that goes up under stress. And then it goes also to the guard cells. So we wanted to check what happened if we first silence ABA sensitivity, both in the bundle sheet and the mesophyll and the guard cells. We do this specific silencing of ABA sensitivity using the mutant of the PP2C, the ABI1/1. I'm not going to get into too much details here, but what we know that if we use the muted PP2C overexpressed in any cells, we actually get a dominant negative expression, which means cells that express the ABI1-1 does not sense ABA in those cells. This is a very convenient way to work because when you work with this overexpression, in fact, you can just simply express the gene and get insensitivity of the specific cell. So Adi Aharon, a PhD student in my lab, and Boaz Nigan, a former master student in my lab, they created cells that do not sense ABA either in the guard cells or the bundle sheet or the mesophyll. I will go quickly because I see that I'm running out of time. So when you just silence the ABA in the guard cells, you get more opening of the guard cells, even under controlled conditions. This is funny when you think about it, because why would silencing an ABA will increase stomatal aperture under non-stress condition? Okay, it was funny because we did not expect to have more transpiration under normal condition. It's like you cut someone brakes in the car and the car drive faster. Should not drive faster, right? It should prevent the breakage. Anyway, when we expose these plants to ABA, we see no reduction in stomatal conductance. This is expected. So these cells should not sense over expression of ABA. Also, when we expose this plant to drought, the stomata was not closed. Although the stomata mechanism was fine, because when we expose the plant to dark or to PEG, the cells work very nice. So this mechanism is specifically sensing ABA. Something which was very interesting is the fact that the VPD response of this plant was very similar. Look at this plant. This is the ABA insensitive guard cells, which look very similar to the wild type. This is the wild type, and this is the ABA insensitive. This is a fusicockin treated plant, but you see the pattern is very similar, only much higher. Again, plants that do not sense ABA in the guard cells actually open stomata wider. So we decided to measure the whole plant. We put the plant on a lysimeter because it's Arabidopsis. We had to put 4 plants in a small pot. Nevertheless, it worked very fine because when you measure the stomatal conductance, the daily stomatal conductance of the guard cells ABI plants, you see a very similar pattern to the wild type, only much higher. And the same thing you see under drought. You have less stomatal conductance, but the ABI opening more stomata. All in all, we come to the conclusion that ABA has a restricting role under normal conditions. So under normal condition, ABA actually prevent the maximum stomatal aperture that could be achieved without ABA. We don't know exactly why plant restricts their stomatal opening, but this is pretty clear. Also, when Boaz expressed ABA only in the mesophyll, now I'm going to show you a few results of ABA insensitivity only in the mesophyll. So this plant do not sense ABA in the mesophyll, but have a normal stomatal sensitivity to ABA. Very funny, this plant was quickly growing and then start to flower. So they were growing faster, but for shorter period of time because the flowering period or flowering time was much faster than the wild type. And as a result, they stopped growing, either in a short or a long day. So this plant flower faster, but as a result has a lower biomass. In fact, the stomata was also wider, although the stomata responds to ABA. You can see it in this figure. This is stomatal aperture. So these are the FA, which mean do not sense ABA in the mesophyll. So they have bigger stomatal aperture, although the stomata work fine. The stomata respond to ABA. Moreover, when you treat this plant to drought, so you find the plant that do not sense ABA in the mesophyll has a very similar yield. We count seed of Arabidopsis as the wild type. Under normal conditions, they are much worse because they flower early and have less biomass. But funny, under drought, this plant has lower biomass, but the same number of seed. So this plant has better harvest index. So to conclude this result, we saw that the ABA insensitive only in the mesophyll actually grow faster, but start to flower earlier, which gives them an escape mode of action, okay? Moreover, this plant has a very huge hydraulic conductivity. Hydraulic conductance. You see that the mesophyll non-sensitive ABA has much bigger hydraulic conductance than the wild type. And similar, the bundle sheet hydraulic conductance is much bigger when the plant does not sense ABA either in the mesophyll or in the vascular system. Again, we wanted to see how these phenomena impacting the whole plant. So again, we use the lysimeter system to measure the whole plant. And here we find something which is really fantastic. Plants that do not sense ABA in the mesophyll actually getting a very big transpiration. So they start normally. You see the first week, you don't see any differences. But as time goes by, this plant completely lose any control on the transpiration. And these Arabidopsis plants transpire about 600 mL. It's like tomato plants, which is much, much higher than the control plants. So all in all, our conclusion from this study that actually under well-irrigated condition, ABA has a very strong restricting role. We don't know exactly why, but it seems that under normal condition, the very basal level of ABA, and always you have some basal level of ABA, this level of ABA actually prevent the plant from reaching its maximum transpiration. Probably it has a very important role in the G2 times E or genotype environment interaction, but we cannot say exactly why it is. But moreover, I want to remind you, we want to study the molecular and cellular level of this ABA in the cells. So what Tanmai did, she decided to measure the ABA insensitive plant using measuring the xylem sub pH of the plants I just showed you. So starting with normal treatment of ABA to the vessels, we see alkalinization. This is the control plant. pH is a bit acidic. Treating the plants via feeding the vascular system with ABA, we get alkalinization, significant alkalinization. When we work with the scarecrow ABI mutant, so this plant do not sense ABA only in the vascular system, we don't see the ABA effect. Again, when we take the AHA2, okay, the mutant that does not express the pump in the vascular system, again, we don't see ABA impact in the vascular system, but when we use the recombinant, sorry, the complemented plant, we do see the impact. So ABA does probably block the AHA2 pump. Moreover, when we measure the protoplast pH, now we go to the cellular level, and we measure the pH in the cytoplasm. So here, we measure the pH here, we get alkalinization of pH in the cytoplasm. Now, this is very funny. If we think that ABA blocks the AHA, so if the AHA is blocked, we can expect to have more proton in the cytoplasm, and we expect to see more acidification in the cytoplasm. So why do we get more alkalinization? When we measure the hyperpolarization, again, activity of the pump, we do get depolarization by using ABA. So blocking the pump does depolarize the membrane. So ABA work as you expect in the depolarization part, but does not work as you expect on the pH level. When we work with the mutant, so the AHA mutant, so as expected, without AHA, the pH in the cytoplasm is a bit acidic, as you expect, because the pump does not work, so you have more proton in the cytoplasm, so it's more acidic. When you put ABA, again, you get alkalinization. And the same thing when you just use a blocker, the same pattern. So the alkalinization of the cytosol actually is not explained by just blocking the AHA. So something else has to be there. And we thought maybe it's the vacuolar pump. So also the vacuolar has a VAHA. It's a vacuolar pump that actually pump proton from the cytoplasm to the vacuole. And when this pump works, it's actually alkalinize the cytoplasm. And indeed, what we saw, sorry that I rush, I see that I'm running out of time. What you need to remember here that when we use ABA plus bafillomycin actually blocked this pump, only then we did not get the alkalinization. So when you block this pump, only then you don't get alkalinization. And when you use a mutant of this pump, you don't get this phenomena. So to conclude this part, what we think is that actually under normal condition, blue light activating the AHA, which throw away proton to the xylem, and the pH goes down in the xylem. We have acidic pH and hyperpolarization of the membrane, both because of the activity of the AHA. This activation actually open aquaporin, which change the K leaf, and the water flux through these cells actually is being motored via the stomatal opening. So the water flux actually being generated via the VPD here. So as soon as aquaporin will be open, you have a very nice flux outside. Under stress, when we have ABA, the ABA actually blocks the AHA2. We could see that. This actually prevents the pumping of proton outside, which alkalinize the xylem. We could also see that. Also, we got depolarization of the membrane. All of this is very logic. ABA also activates the VAHA, which pump proton into the vacuolar and in fact alkalinize the cytoplasm. This alkalinization of the cytoplasm basically close aquaporin. This was shown in Nature paper, that actually all the aquaporin sensing the pH in the cytoplasm Closing of aquaporin, of course, reduce the K leaf and in fact reduce the whole water conductivity of the leaf. Obviously, we have a lot of things to still study in this mechanism, and this hopefully will be done in the coming years. Lastly, I will not go into details because we don't have more time, but I just want to mention that by in the transcriptome of the bundle sheet, we also find chitin and flagellin receptor. These MAMPs actually are also very known in the guard cells to sense chitin. Chitin is a molecule that found a lot in the cell wall of fungus. And this chitin is usually released in the xylem much before the fungus is well spread in the plant. So you can say that you find chitin in the vascular system even before the actual fungus reach these cells. So we found these receptors in the bundle sheet. These receptors are known to involve in disease, in fungal diseases. And Ziv Attia, a former PhD in my lab, now doing his postdoc in Colorado University, he could actually show that both mesophyll and bundle sheet actually reduce their water permeability in response to chitin, and also the vascular system was reducing its K leaf. He proved it in a nice paper recently published in Plant Journal. I'm not going to get into details, but the conclusion here was that when the plant sensed the attack of fungus much before the fungus reached the actual cells, the leaf actually cut the water supply to the leaf. So we could see isolation of the whole leaf in response to the chitin. We also find the BAC1 and FLAS2. These are flagellin receptors. So these are bacterial receptors. Again, these receptors found in high levels in the bundle sheet. And when we look on the flagellin receptors, so Ahan Dalal, postdoc in my lab, going to finish very soon, his postdoc. So he showed that when you use flagellin, you get, again, reduction of the hydraulic conductivity and osmotic, so both osmotic conductivity and hydraulic conductivity of the mesophyll and the guard cells. And what we concluded here, that when the plant is under attack of bacteria, the flagellin that released from the flagella of the bacteria actually reduced the water flux from the mesophyll cells to the cell wall. And when the bacteria try to establish on the cell wall, the blockage of the aquaporins here actually make the water potential in the cell wall to be much more negative, which actually inhibit the bacteria chances to establish on the cell wall. So this is a mechanism we just suggested. It is now under revision in Plant Journal. So this is the mechanism we suggest, that even under biotic attack, the vascular system and the mesophyll system has a very important role to actually protect the plant from the attack. So all in all, in conclusion, the take-home message that I want to give you is that usually we treat the vascular system as a dead cells. We often teach this even. Okay, xylem is dead cells. This is obviously true. But in the more than a decade, when I'm studying this bundle sheet, I tend to look today on the vascular system as the bundle sheet. So I look at the vascular system on the pipe as the living vascular system, and the xylem, to my opinion, is only a scaffold. It's only holding these membranes in tight. So this actually, to my opinion, these cells are actually the pipe, the smart pipe that regulates water and ion going out of the vascular system. So looking at the vascular as a dead cells is not completely true. Finally, so I did acknowledge all the wonderful student and collaborator I was lucky to work during the years. And finally, I just want to acknowledge ISF, which through all these years fund my research and still does, and the Ministry of Agriculture, that some of the results you see here was also part of his result, and sorry, his fund, and thank you for your attention.
Thank you, Menachem. was a comprehensive presentation for a long period, and I think it's open for questions now. So please do. Okay.
Menachem, can I ask something? Hi.
Hi, Joseph.
Thank you. It was very clear. My question is actually going from where you stopped to where the water actually is leaving the leaf as gas. Can you say something about the pathway of the water from the liquid phase in the cells to the gas phase in the intercellular space?
Yes. So this is the place. When you look on the mesophyll cells, okay, this cell wall, what you see here, this is the place when water leaves the cell wall and becomes vapor. So the matritic potential in this part is actually constructed from the cell wall. Now, what is wettening the cell wall is actually the membrane just below. And aquaporin in this membrane, which actually transport a lot of water. So the water going from the cell to the cell wall will be first wettening the cell wall. So if you actually reduce the membrane water permeability, you will get this high water potential in the cell wall. So you will have much less gas evaporation than you will have under wet cell wall. This is because of the matric potential of the cell wall. So as a result, the whole water potential of the whole leaf will go down and stomata will be closed. So the cell wall has a very important, very, very important role as a transducer of the water in the cells and the water potential in the leaf. It's exactly a transducer converting water content to water potential. I hope I was clear here.
Thanks.
Menachem, may I ask a question?
Sure.
Or actually make a note. First, great work and very nice presentation of it. You've mentioned the measurements of pH within very well-defined compartments of the cytosol and the vacuoles and so on. And I'm not sure I followed all the details, but you pointed out to some abnormalities in this sense. And here you may have another explanation, which is kind of local proton concentration, which is well known from the, I think I'm cut off of power.
We can hear you, Tzach.
Tzach?
Okay. אוקיי, צריך כנראה עובר משרד, אם יש למישהו שאלה בינתיים עד שהוא יגיע למקום עם מחשב.
אני יכולה לשאול, מנחם?
בבקשה.
היי מנחם.
אהלן.
זה היה ממש מדהים. I'm sorry, I need to speak English, sorry. It was fascinating, thank you for this talk. And I want to ask you, this phloem is also included in the bundle sheet area inside, next to the xylem.
Right.
Do you think there is also a role for the phloem cells in these mechanisms?
Actually, I'm pretty sure. I want to show you an amazing picture. This one. You see this picture?
Yes.
From Doring et al. This is the bundle sheet. You see? So these are the xylem and these are the phloem. You can see actually. And you see that like half of the xylem is covered with bundle sheet and all the xylem actually touching a bundle sheet. There is no xylem that do not directly touch a bundle sheet, But for the phloem, you have the companion cells and other cells. So clearly there is some role here, but someone have to find it.
Okay, let's talk about it. Okay, thank you.
Okay. Right. So it looks like that there are no questions. From Zach, sorry, the computer is okay. Zach mentioned that the computer lost power. Okay.
I will
think
catch
you
them in
can.
the corridor.
I think you can find you. We can talk. So thank you very much, Menachem. Our next seminar will be on April 4, 9 a.m. by Jose. We'll have an invitation sent in a few days, but this is just a reminder. So thank you very much, everybody.
Itai,
Ken?
is the recording available later?
I think it would. It will be online, and then we should put it somewhere. We have to check where, but it will eventually be available.
Thank you.
Okay. So thank you, Menachem, and thank you, everybody.
Bye-bye.